Good day, everybody. I think it's about time for us to get session 4 begun. And I want to thank everybody for joining us, no matter what time of day you're in your local area right now. So I think this is a really exciting session, a good continuation of the meeting that we've been having so far. This one focuses on, we have an interesting set of speakers who are going to be talking about how to integrate curation, how to integrate automation into curation workflows. So we begin with Dr. Sheng Wang. He's an assistant professor at the School of Computer Science and Engineering at the University of Washington. His research interests include biological NLP and computational biology, and he's here to talk to us today about precision curation, generating sentence descriptions for new biological discoveries. And a reminder to all of the audience to use the Q&A area for questions, and for my speakers to please set a timer for yourselves. And Sheng, go ahead and take it away.
Great. Can you hear me now? And can you see my screen?
I can hear you just fine. Yep.
Okay, great. Good morning, everyone. I'm Sheng Wang. I'm a new assistant professor in the School of Computer Science at the University of Washington. Today, I'm going to introduce a new curation paradigm called precision curation. This is an analog to precision medicine in modern biomedicine. So let's first take a look at some issues in the current curation annotation paradigm. For example, we have a single cell data set, and we want to annotate the cell type for each cell here. We first cluster this data set. Then for each cluster, we find one term from a predefined set of controlled vocabularies. For single cell, this controlled vocabulary is from the cell ontology. For other problems, we have other controlled vocabularies or other sets of ontologies. So this annotation step is often the first step before we analyze the new data set. Controlled vocabulary are very crucial here for us to do the annotation, but they could have some limitations. The first issue is that they cannot annotate new discovery. So new cell types are not collected in existing controlled vocabularies. New diseases like COVID, they are not collected in disease vocabulary like MASH when COVID just started. Secondly, controlled vocabularies may not be precise. We don't have enough controlled vocabulary to support the annotation of a very large data set. For example, here, many cells are annotated into microglial because they are in this orange cluster. But the cell here and the cell here, they could be very different. They could have very different gene expression vector, but they are still annotated to the same ontology term. Lastly, controlled vocabulary may not be expressive enough. They are often a short phrase of 1 or 2 words, so they could be ambiguous. And these 3 limitations actually become worse in large data sets and when we have new technology in the future, because we will make new discoveries that may not be annotated by existing controlled vocabularies. So our solution to address this kind of problem is motivated by the success of precision medicine, which essentially provides a personalized treatment to each patient. So here, we want to do a precision curation for each data instance, not a cluster. For example, here, a cell in a single cell data set, we want to provide one annotation in the form of sentences. So cells that are similar will have semantically similar sentences, but the sentences will be different for 2 different cells, even if they are in the same cluster. So we actually speak in sentences when we communicate. For example, when I give this talk, I talk in sentences, not controlled vocabulary, right? So I think sentences can help us address these issues from the controlled vocabulary. First, controlled vocabulary cannot annotate new discovery, but we can generate sentences on the fly to describe new findings. Secondly, controlled vocabulary may not be precise, and we don't have enough controlled vocabulary that is scaled to very large data set, but the space of the sentences can be scaled to the space of new biological data sets. Lastly, because sentences are much longer, so they could be more expressive than phrases that are often used by controlled vocabulary. So here, we don't want to replace controlled vocabulary with sentences. We think they supplement each other, and if we use them together, we can better annotate a biological data set with a better resolution. Our research focused on generating sentences to describe a biological data instance, not a cluster or a data set. And here we develop a machine translation model. So our machine translation model does not translate from one language to another language. Actually, our machine translation model translates from one biological data instance. For example, one node in the ontology, or a protein sequence, or a gene expression vector, or a pathway. So we translate one such biological data instance into human text in the form of a sentence. And this is how we do the curation or annotation. Our method is supported by large-scale language model, which has recently dominated the NLP domains. The language model we use here is the PubMed-BERT, which is trained specifically from millions of scientific papers from PubMed. And this kind of language model can help us utilize the existing literature, existing scientific literature, to build this machine translation model from biological data to human language. Today, I'm going to talk about two of those applications here. The first is to generate sentence definition for a given terminology in the ontology. So these terminology are used as control vocabulary in existing curation paradigm. Biologists have spent lots of efforts to curate the definition for those biomedical terminology and accumulate a very good data set for us to build a machine learning model. Now we want to use machine learning approaches to automate the generation of this terminology. So the generation of the definition of those terminology. So the input of our model is a terminology, a phrase. And then we want to train a model, a supervised learning model, to output the definition, the sentence definition of this terminology. So for this terminology, we do not know its definition, but we require it to be within the ontology graph. And on this ontology, we know the definition for some of the terminologies, and they will be used as the training data. And we want to generate the definition for the remaining terminologies. The key idea of our model is to use the nearby terminology on the graph. So we want to use the information of neighbors on the graph, their terminology, and their definition text to help the generation of a new terminology. And this is a graph-based approach to solve this problem. So this is our machine learning model. Here, DI is the text, is the definition we want to generate for a new terminology, and it is conditioned on three kinds of input. The first input is the embedding of the terminology. We see a terminology, we want to generate definition. And this embedding is generated using a BERT language model. So here we use PubMed BERT here. The other two features are based on the graph structure. We first use a graph neural network to propagate all the terminology over the graph, and then we can get a graph smooth embedding for the terminology we want to generate the definition. We also use another graph neural network to propagate over all the training definition over the graph to get a graph smooth embedding based on neighbors definition. So these three features are used together to train a neural network and finally put it into transformer to generate the definition for a new terminology. We compare this method to existing text generation model in NLP, for example, transformer, sequence to sequence, and the conditional VAE. So these approaches do not consider the graph structure. We found that our method achieved much better results, and those metrics measure the n-gram similarity between the generated text and the ground truth text. We also perform an ablation study to study whether those graph structure are important. So we found that the terminology graph and the definition graph are both very important to sentence generation, and they can help us generate better definition for a new terminology, as long as they are in the existing, as long as we put it into a position in the ontology graph. So here are some examples of sentences we generated. First, those are sentences we generated. First, we can see that the sentence generated by our method is quite similar to the ground truth sentence. The reason is that we effectively utilize neighbors definition or neighbors terminology on this graph. So you can see that those sentences also look similar to the parent or child node on the ontology. We also did some error analysis and noticed that some of the key entities generated by our method could be incorrect. For example, in the ground truth, it should be a tumor in the eye, but our generation actually output gastric tissue, so tumor in the gastric tissue. I think this is because we generate gastric when we see the word tumor in the terminology, and BERT language model try to connect tumor with gastric, which is with gastric in their language model parameters. So the overall sentence looked good, but the key entity could be incorrect. So in the future, a very important topic we want to study is to incorporate or have human experts to help refine it and put human experts into the AI loop. We want to develop an AI method to also further refine the key entity. And this will be a collaboration between a human expert and the AI model. So the second project I want to talk about is a pathway description generation problem. Here we have a pathway. This is a pathway graph structure. We have different node types, different edge types. For each node, we have a sentence description. Okay, so each node in this graph, we have sentence description, and this description has nothing to do with the graph structure or the neighbor in the pathway. It's just a description of this node. The goal here is to generate one description for this pathway. So this is the description we want to generate, and this problem can be regarded as a summarization problem. We want to summarize this pathway based on the sentence on each node with the consideration of the graph structure. The key idea of our method to do this is to convert this pathway to text or graph to text problem to a text to text problem. The reason is that text to text problem takes a text input and output another text. This problem setting has been widely used in machine learning and NLP. So existing NLP techniques and language model can be applied to text, but not graph to text. How can we convert this graph to text problem to a text to text problem? We use a k-nearest neighbor based approach to do this. We first find neighbors, similar neighbors with similar graph structure and similar node descriptions. And then we concatenate the neighbor's description into a very long meta description. Then we train a summarization model from this meta description to a concise description for this pathway we are interested in.
Excuse me, Sheng, I'm sorry to interrupt, but you have about 2 minutes.
Okay, yeah. So this is the experiment result on PathBank dataset. It has thousands of pathways with their curation description, and our method achieved the best result compared to the graph neural network and transformer. In the future, we want to incorporate non-text node features into the model, such as protein sequence and chemical structure into this framework. So this is all about my talk today. I have talked about the idea of generate descriptive sentences to overcome some existing issue in using controlled vocabulary. Those sentences can annotate new discovery if we generate on the fly. They are also more expressive and can scale better to large data sets. I have demonstrated some results on terminology definition generation and also pathway description generation. We show how language model and transformer can generate good templates, but they still need refinement from human experts. In the future, we would like to build a human-in-the-loop precision curation paradigm to further improve this generation. And in this loop, AI model will generate sentences, even not a perfect generation, but still provide templates to help experts accelerate their annotation. And this new annotation can be later used as training data to further enhance the AI model. Thank you. This is all about my talk, and I'm happy to take questions. And those are the papers I talk about in today's talk.
Okay, thank you so much. Really appreciate your presentation. A lot of interesting information there. I do see something in the Q&A session. It's perhaps more of a comment. You're in one of your early slides where you had clusters and you highlighted, that cells on one end of a cluster might be something a little bit different than cells on the other end of the cluster. The comment I think relates to that slide. I'll read it now. It may not be a different cell, but a cell at a different stage of its development or life cycle. And so the SC transcriptome data is on cell type, but not necessarily on developmental stage. And so the question, I think, is really, does that mean that there are deficiencies in this system?
Yeah, I agree. So this is a great question. Actually, we not only want to annotate cell type, but also may want to annotate cell states. For example, we can use control vocabulary to describe the cell type, T cell, but maybe we can use sentence to provide more information about the cell states and what stage this cell is in. And this could be a hierarchical model where we have control vocabulary from general cell type to specific cell types. And then from specific cell type, we go to detailed sentences to describe the states or maybe the different overexpressed, underexpressed genes in that cells to provide a better overall picture of the difference within this cell type. So this is our thought here.
Okay, thank you so much. Warm round of virtual applause to our first speaker. I don't see any other questions for you at this time. So, and you're free to also write a response in the Q&A session if you want to do that. There is a request to share a link to the graphene paper, and you can just do that in the chat area. So I'd like to move on to our second speaker, which is Dr. Lynette Hirschman. She is a MITRE Technical Fellow and Chief Scientist for the Biomedical Informatics at the MITRE Corporation, leading research at the intersection of biomedical informatics, computational biology, and natural language processing. She is a founder and co-organizer of BioCreative, an international challenge evaluation for text mining in biology. Lynette is here to talk to us about BioCreative today. And Lynette, go ahead and take it away.
Great. Well, thank you so much for the opportunity to present here and for a great workshop. It's been really fascinating. So I'm going to talk about BioCreative. This is going to be a lightning tour through many years of history with a focus on assessing text mining tools for the biocuration workflow. I want to begin by acknowledgements because those always get left to the end, and really BioCreative has been the work of a great many people over the years, and I want to particularly call out some of the people you've already seen and heard from, Alfonso Valencia and Martin Krallinger from the Barcelona Supercomputing Center, the great work and contributions from the NCBI team, Jiang Lu, Rizarda, Islamai, and many others, and also the contributions from Kathy Wu and Cici Arrighi at the University of Delaware. I should also add that BioCreative would not have happened without close collaboration with biocuration teams, and in particular, the system developers. We must have had several hundred of them by this time, and without them, BioCreative simply wouldn't have existed. So with that, I'm going to move on to tell you about BioCreative, which is critical assessment for information extraction in biology. And this is collaborative, as I just said, international and multidisciplinary. We organize challenge evaluations and workshops that address the needs of the biocuration and bioinformatics communities. And you can see off in the bubble to the left there, some of the biocuration teams that we have worked with over the years. And we develop resources and standards, all of which are to facilitate putting text mining tools into the curation workflow. So we have been doing this for quite a while. Our first BioCreative started in 2004. It was held in Granada with Alfonso Valencia and his team hosting it. And in November of 2021, we held BioCreative 7, which was a virtual meeting, but hosted by Cici Arrighi at University of Delaware. On top here, I've put down some joint workshops that we've also done with the biocuration community in ISB. We've had joint meetings, we've had workshops, and these have been very important in getting curator input. All of this is focused on dealing with real users, so the biocuration community, their real problems and data, curated data, using community standards. So I'm going to talk very briefly about the three kinds of things that we have done. One of them is to evaluate tools. And there you see a little screenshot from a paper from BioCreative 2, where one of the tasks was to have the systems extract lists of gene identifiers associated with 3 of the different curated model organism databases: fly-based, mouse, and yeast. And if you look closely, you can see that the yeast results are pretty good. This is a precision versus recall graph, so you want everything to be up in the upper right-hand corner there. And mouse and mouse and fly were significantly harder for the tools to do. I'm then going to talk about curator needs, and that's a little graph of the curation workflow that we put together out of a workshop we held with the BioCuration Society. And the last thing I'm going to mention is building infrastructure. This is a little picture of a meta server aggregating information from multiple annotation servers. That tool was put together, or that system was put together by Florian Leitner et al. From Alfonso Valencia's team. So here are a few examples of BioCreative challenge tracks, just to give you a flavor of what we do. We've already heard a lot about triage in the preceding session. So the first thing that a curator needs to do is find the articles that she or he wants to curate. So we have done some tracks for example, gene ontology curation, identifying articles with curatable Go information, and tracks on protein-protein interactions, identifying the relevant articles that contain protein-protein interaction information. These are generally done on abstracts because of the volume. Then you can go to full curation once you've found the papers you want to curate. And the process there involves entity detection, so finding the major bioentities like species, genes, proteins, diseases, chemicals, and drugs, and then the relationship among these, including evidence for those relationships, so that the curator, for example, can quickly look at something and decide whether it's relevant or not. Examples of these tracks that we've run in the past, we work closely with the comparative toxicogenomics database team to identify genes, chemicals, and diseases linked to the CTD vocabulary. We have done protein-protein interaction curation, actually several rounds of this, extracting the interacting pairs of proteins and the underlying method and the evidence for that method. And a track that Martin Krallinger started a while ago was the chemical drug named entity recognition track, where you extract names of chemical entities, and that has gone on to do much more complicated things with a very large attendance and large number of participants. So moving on to the work we've done in trying to understand curator needs. In 2009, we participated in a workshop, text mining for the biocuration workflow at the 3rd International Biocuration Conference, and wrote up an article which is referenced below. We surveyed respondents from 23 databases, 30 respondents, and discovered that 70% of the biocurators at that time had tried text mining. A little less than 50% were using it, and the most widely used system was Texpresso. They also provided input in terms of the top priorities of tools that they wanted or capabilities. So triage, we've already talked about, identification of evidence in text, and linkage to biological nomenclatures, taxonomies, and ontologies is extremely important. In addition, they wanted access to the full text. It's interesting that we heard in Patrick Rusch's talk earlier about access to supplementary materials. They also wanted that even back in 2009. Also, curation of figures and tables was highly valued. In 2017, we did the same thing again, having a workshop on biocuration and the research life cycle, organized by Cici Origi and Rizarda Islamai, just to mention 2 of the people you will have heard from. We did a survey from 80 workshop participants. 50% had used text mining, and you will notice that's not a big increase from 2009. And of those, 60% were satisfied or highly satisfied with those results. It's interesting to look quickly at some of the comments. So satisfying was the tool was integrated in the workflow, and we really have put a good deal of thought into that. Satisfying was able to use for triage or used for Go cellular component annotation. Neutral comments no quicker than using PubMed. And the information needed is contained in figures. And finally, unsatisfying things were gene name errors and detection of irrelevant interactions. So it can't be noisy or they won't use it. Last, I'm just going to do a flying tour through the infrastructure area. And I pulled out one example from BioCreative 5, which was to create a BioCurator assistant for the BioGrid curators. And that was done using the BioC standard, which had been developed in a previous BioCreative. That's a data interchange standard, and that allowed us to assemble a pipeline for curation integrated from 9 different teams and 8 different components, which are shown off in the graph to the little image to the right there. Those different components did different things, although some of them also did the same thing. So we had components for gene, protein, and species extraction and normalization. We had components for protein-protein interaction and extraction of gene interaction and the evidence passages associated with that. And those were all aggregated and presented through a visualization tool for the curator to actually use. So that was a demonstration of interoperability and interactivity supported by standards. So where are we? We have been successful. We've held 10 workshops with over 40 evaluation tracks. The most recent BioCreative 7 had 5 tracks with 88 teams participating. We have built a community of biocurators, bioinformaticians, and computer scientists. We've established benchmarks and standards, and in particular, reusable corpora. And if you're interested in the corpora, if you go to www.biocreative.org and click on the resources tab, you will see the corpora that are available through that website. And we also have demonstrated uptake of tools. PubTator is one of the big successes used by Uniprot. TMVAR also used by Uniprot. TagTog has been used for gene indexing in Flybase. Odin has been used in PharmGKB and the Regulon database, and RLIMS-P has been used in phosphogrid curation, and all of those had participated in BioCreative at various points and leveraged some of the resources from BioCreative. So what's next for BioCreative? This workshop has already been a wonderful place to understand the state of the curation community and the state of the community developing tools for text mining and resources. I'm curious to find out whether there is more widespread adoption of text mining, since that is clearly, as Andrew Su just mentioned, a way of increasing the scalability of curation. And also, I'm interested in identifying the priority unmet needs of the curation community. We also are looking at technological gaps. So, for example, how to quickly adapt generic tools to new applications. This is a major problem. As we produce better generic tools, each curation workflow is different, and the criteria for curation are different. So each tool has to be adapted to its new application, and you don't want to have to train massive numbers of, create a new curated data set in order to retrain your tool using machine learning. It's important to understand how to partner, how humans and the tools should partner to create tools and interfaces that are good enough that curators want to use them. And we are also very interested in identifying new applications and new uses. For example, in BioCreative 7, one of the new areas we tackled, Graciela Gonzalez and her team at UPenn had a track on curating Twitter feeds for mentions of drugs. And that was really interesting. We're also interested in getting more into the medical area. In the US, at least, that community is very fragmented. There's a completely different set of people who are doing text mining for, say, clinical records. But anyway, we are interested in seeing what we can do there, and also had a panel on extracting adverse drug events. So we would love to hear from you and have you work with us to formulate the next critical biocreative tasks. And with that, I thank you and we'll take any questions.
Thank you, Lynette. That was a really interesting talk. I'm going to have to learn more about biocreative products myself. So there is a question for you. Do you invite researchers and scientists to your meetings, or just the biocurators and developers that you have established relationships with?
Absolutely. We invite, in particular, we invite the developers are, of course, a critical part. We do invite researchers and scientists. We often have invited talks. For example, Josh Denny gave the invited talk at BioCreative 7. But we have trouble attracting sort of people with more generic interests because this is a fairly specialized kind of area.
So then how do you, here's another question. This one comes from Helen Parkinson. She's telling you, great talk. Her group uses text mining, and as you said, there are variable workflows and these change rapidly. I wonder if you have success stories or experience of portable NLP components plugging into existing interfaces and workflows.
Great question, Helen. It's one of the things that I've been thinking about in terms of actually trying to develop some good challenge evaluations around. Unfortunately, at this point, it's still quite labor intensive to do one of these evaluation challenges, especially if we want to have sort of, statistically significant results to report out. So there are sort of two parts of this. Evaluating interactive systems is very tricky in general, and figuring out how to evaluate rapid tailoring to new domains, I think, would be a really interesting challenge, and I would certainly like to, think about it and talk to you some more about it.
Okay, so thank you so much, and you can follow up with your answers in the Q&A session also.
Great,
so I'd like to, give Lynette a round of applause and move on to Dr. Alan Bridge as our next speaker. Alan is director of the SwissProt group at the SwissProt Institute of Bioinformatics. He is a co-principal investigator of Uniprot and principal investigator for the RIA knowledge base of biochemical reactions, the enzyme resource for enzyme nomenclature, the Swiss lipids knowledge base for lipids and lipidomics, and the ProCite and HAMAP resources for protein classification and annotation. And Alan is here to present a talk on expert curation at scale in Uniprot KB SwissProt. Alan, take it away.
Okay, thanks, Kim, for the introduction. Can you hear me okay?
Yes, I can hear you.
Thank you. Thank you. So I'd like to begin by thanking the organizers for putting together a fantastic workshop. It's been a really interesting couple of days. And I'd like to spend the next 12 minutes or so sharing some of the work which we're doing in my group to try and accelerate the curation of information about protein functions actually into the Uniprot resource. So given the time limitations, I'm going to focus on one very specific aspect of the biocuration that's done at Uniprot, and I'm going to be focusing on enzyme functions. And there's a couple of reasons for that. I mean, we've done a lot of work in the last couple of years to try and improve the representation of enzyme functions in Uniprot using machine readable ontologies and other standards. And I hope to show you how that can be used as a base to try and train machine learning and other methods to accelerate the extraction of relevant information from publications. And just following on from Lynette's talk, I think this would be a really good subject for a biocreative evaluation, and it's something we'd be really interested to talk to the biocreative folks about. So my team is involved with collaborators at EBI and PIR in the development of the UniProt resource. Within UniProt, we curate protein sequences and information about their functions using a broad range of terminologies and standards, like the gene ontology, for instance, for functions and processes, IMEX for molecular interactions, ECHO for provenance, that's the evidence and conclusions ontology, and RIA and KEBI for chemistry. In addition to this curated core of knowledge within UniProt, we also have a large body of computationally annotated protein sequences. Obviously, computational approaches which infer protein functions by homology are a great way to scale curation, but I'm not going to be talking about those approaches today. I'm going to be focusing really on the curation of proteins and specifically enzymes. So within UniProt, we use a resource called RIA as kind of our reference vocabulary for capturing information about enzymes and transporters. Like UniProt itself, it's a curated knowledge base. So it's a standalone resource as well as a vocabulary, a bit like the gene ontology, if you like. It's built on a chemical ontology called KEBI, which is developed at EMBL EBI in the group of Andrew Leach. So KEBI provides the building blocks for RIA, and the reaction data is provided in a variety of machine readable and human readable formats, like for instance this graph representation and other representations which can be derived from it. We're now using RIA to curate UniProt KB. There's around 40% of all SwissProt records curated with this vocabulary. So this provides a basis for developing methods to try and improve the scaling of enzyme annotation. So basically what we want to do is we want to identify literature about enzymes, extract functions from that literature, and then use that information to populate linked resources. So as well as UniProt and RIA, we're also working with the gene ontology and Reactome to try and align the representation of protein functions and enzyme chemistries in these various resources. So all the work we're doing for UniProt and RIA will percolate through into these other resources too. So to give you an idea of the scale of the problem, we've built a variety of classifiers to investigate this problem. We use a resource called LitSuggest. Lynette mentioned some of the tools that are being used at UniProt. LitSuggest is really one of the latest additions to the toolbox. It's developed by Ziyong Liu's team at NCBI, and it's really a fantastic resource for building classifiers. So no knowledge of machine learning is required. All you need to be able to do is to build a good corpus in order to build an effective classifier. So we were charting the task of enzyme curation using LitSuggest. We built a classifier which is capable of identifying literature about enzymes, and we use that classifier to scan the entirety of index abstracts. And we found about 1% of published literature deals with enzymes or transporters. Okay, so this is using quite a strict threshold. We also looked at the problem in other ways too. So we tag chemical mentions within those papers using a tagger called Bluebird, which is part of the NLM Chem tool set. So we map chemical mentions to entities in MESH and the chemical ontology KEBI, so we can extract chemical structure data for them. We can then calculate chemical fingerprints, and we can build a map of all the chemical mentions, which are potential substrates and products in the literature about enzymes, and that is what you see here. So the parts of this map in red are parts of chemical space which are not yet curated at all in RIA or UniProt. And the parts in blue are at least partially curated. We have at least one reaction, one enzyme function, which includes or concerns one of these chemical mentions. So this gives you an idea not only of the scale of the problem, but also something about the distribution of the problem. So there are large parts of chemical space which are still to be visited, and some parts of chemical space have been pretty well curated. And this is a tool that curators can use to explore the curation problem. They can zoom in and analyze interesting parts of chemical space. Here, for instance, we have a drug which is used to treat diabetes and a bunch of related compounds around it, which all need to be curated. So this gives you an idea of the scope of the problem which we're addressing. You know, as Andrew mentioned, it's difficult to scale professional curation. I think at this level, it's probably also difficult to scale community curation as well. And I think we definitely need better computational tools to address this kind of problem. So one of the approaches we've been looking at Swisprot and with collaborators is using language models to try and address this problem. So we're not an NLP group. We're not developing methods to extract information from text or to tag information from text, but we do know a bit about chemistry, and we are developing some tools which could be incorporated into NLP pipelines. And this harks back to, I think, the comments again of Lynette about incorporating existing tools into NLP pipelines to address new problems. So here we're using language models. So the idea is you can express chemical structures in kind of language notations called SMILES, which is shown here, where you can think of atoms as being like letters and the molecule is kind of like a word. And you can then use language models to do operations on these chemical structures or these proposed enzyme functions. So we heard about BERT models from some of the previous speakers. So BERT is a language model and it can learn about dependencies in language. And you can also teach these models too about dependencies in chemical structures. So what makes a valid chemical structure, what makes a valid chemical reaction, and how atoms in structures, in substrates and products relate to each other. So you can build classifiers that are capable of identifying true substrate product pairs from the text just based on their structure and without even using the language. And this kind of approach of language modeling for chemistry has been pioneered by a number of groups in Switzerland, including Jean-Louis Raymond in Bern, Teodoro Leno at IBM, and Philipp Schweler at EPFL in Lausanne. So this is one approach that we're trying to address this problem. So it's a tool that we'd like to plug in really to existing NLP pipelines. So the training is kind of a classic transfer learning process. We have a general model of reaction chemistry, which is derived by training on a very large database of text mine data from patents. And we then retrain on a much smaller data set of substrate product pairs, which have been curated in RIA. So it's something like 13 million text mine reactions from patents, which are a little bit easier, I think, to manage than biomedical texts, and a much smaller set of positive and negative pairs, which are drawn from RIA and KEBI. The performance of the model after it's been retrained is pretty good. It acquires the ability to distinguish pairs of substrates and products just from random pairs with pretty good precision and excellent recall. So we're now testing these models on actual enzyme literature. So we take all the abstracts from our LitSuggest scan. We treat each abstract simply as a bag of words or as a bag of chemicals. We extract all possible pairs of substrates and products from the abstract, and we then classify them using the BERT-based model of chemistry. Again, the precision is pretty good and the recall is excellent. This is really, very preliminary data. Actually, we obtained this data last week, and we've been frantically curating the data over the weekend. But the model seems to perform pretty well on real data that it's not seen before. So we think this is a promising approach, and that these kind of language modeling approaches for chemistry could be adapted for these kind of tasks in biocreative and other evaluations, and also in production pipelines as well for NLP. We also managed to recover a lot of the data that's been curated into RIA over the years. About 1 in 3 of all of the known substrate product pairs from the RIA dataset could be recovered from abstracts using this approach. So it seems quite promising. So this is an example of one true positive prediction that the model obtains. We have a small part of text which talks about 3 chemicals. For those 3 chemicals, we create the 3 pairs and we scan using the model, and the model correctly identifies the main pair of reactants from these 3 pairs. So it seems to have some potential as a tool for NLP. Obviously, this is quite preliminary work, as I said. We've been working on this kind of approach for a few months. There's a lot we could do with this. I think one obvious thing is to plug this into existing NLP pipelines. So to combine with language modeling approaches for language. So to consider the structure of the text when trying to extract the pairs. There's other things which we can do with these kind of models, which one can't do with classical NLP approaches. So we can derive generative models. So the idea here is that for each chemical mention, which you find either in a database or text, you can predict what would be the possible precursor or the possible product, right? So you can generate hypotheses which you can then search for in the literature. And this is something that we intend to test with this model. And obviously there's a lot of data issues to address. We can augment the training sets by writing the chemical structures in different ways. This is called data augmentation. There's a lot of room for improvement in the way we process the training and test data. Stereochemistry is an issue and things like charge is an issue. And obviously we'd like to test on the full text eventually. Just a couple of final thoughts. These models are, I think, potentially very powerful, but they do require very, very high quality data, I think, in order to get good results. And our experience of working with this kind of approach is that we really need the expertise of curators and domain experts in order to generate good models. Obviously, all of this work ultimately depends on curated data, resources like KEBI and RIA and MESH and others. And without those, obviously, these systems couldn't be built. And I think really underlines the central role that curators have today in biomedical research. Expert curation is very much, I think, an enabler of these kind of so-called AI technologies and is central to data science as a whole. Lastly, just in response here, again to something Lynette said, we'd be very interested in participating and helping organize a biocreative task in order to develop methods to do this kind of enzyme function extraction from text, basically chemical entity recognition, and then finding relations between chemical entities in papers is what we'd like to do. So I'd like to acknowledge all the people involved in this work. Blanca Cabrera Gil is the data scientist who's working on this in our group, aided by Anne Morga. She leads the RIA project, and Venkatesh, who is the lead developer, and Christian and Elizabeth, who helped curate the data sets for training these models, and Nicole is our head of software development. I'd like to thank Ziyong and his team. So we work a lot with Ziyong on projects like LitSuggest. We did a lot of work piloting this in Uniprot and on other projects like PubData and Bluebird. It's been really crucial working with Ziyong on all of this. And I'd like to also acknowledge researchers at IBM in Zurich who make all of these chemical language models freely available, or at least the ones which are trained on available data sets, freely available data sets. I'd like to thank also all the people at Uniprot. Ultimately, the goal here is to populate the Uniprot resource. Uniprot is a collaboration between ourselves, SIB and EMBL EBI and PIR, and also obviously to thank all the funders. RIA is supported by CERI in Switzerland, and Uniprot by NIH, EMBL, and also CERI. And with that, I'd like to thank you all for listening, and I'd be happy to take questions.
Thank you very much for that really interesting talk. I have a question. So as automation is of growing interest to a number of different repositories and data sets, including those that are curated gold standards, to what degree would automation of applying, for instance, mesh terms negatively impact the kind of work that you're trying to do? This is a hypothetical question.
So in this instance, it wouldn't be too much of an issue. So we extract the pairs using named entity recognition tools. So the actual tagging is done automatically. So we're not using necessarily the annotations of mesh. Did I understand your question correctly?
Oh, I was just trying to understand if that was a risk factor, if some of the underlying databases that fed into systems as being gold standards
Yeah.
were not fully manual. They did not have like full manual curation.
So at least for our application here, what we're trying to do is to accelerate, but not completely automate. So what we will extract from the data is, from the paper, sorry, is kind of pairs of substrates and products, and we'll then curate them into the data set. So we always maintain a separation between the curated gold standard and the additional data, a little bit like UniProt has Tremble and SwissProt. I think as long as you make a good, a clear distinction between those two data sets, then incorporating automatically annotated or automatically curated or derived statements or assertions shouldn't be an issue. But we definitely need, I think, to scale better the extraction of kind of preliminary assertions.
Okay, thank you. There are two other questions in the chat. The first one, I'm a little unclear on how enzymes and chemicals are overlaid. Are just the enzymes from UniProt tagged when Bluebird identifies them, or is there a different workflow? Or another?
There will, yeah, so we don't tag, we're not considering the enzymes at the moment. We're just trying to extract the functions, so just the chemical pairs. So on top of that, we will later extract the enzymes for each of the chemical pairs.
Okay,
but for the moment, yeah, in this particular workflow, it's all about the chemistry.
Okay. There's a second question. Has this model approach been used to build a feature search library or for unknown chemical structures? What comes to mind is unknown lipid species. I feel unknown lipid species data are often left out of publications because there are no identifiers mapped to the structures, where those unknown structures could be uploaded into a text mining database for potential search matches.
Yeah, that's an interesting question. So there's been a lot of work on using these kind of generative models for building unknown chemical structures or enumerating all possible chemical structures. We've also actually built a library of possible lipid structures using KEBI. So the way we did that was to enumerate it using building blocks of lipids, known building blocks. So that number's around 700,000 structures and probably covers most of the possible masses. We didn't use these kind of language models to do it. We used simpler approaches by combining smiles or fragments of smiles. But this is something that's been explored a lot with these kind of models. But
Okay.
not to my knowledge specifically for lipids, but for general chemistry, yes.
Okay, thank you very much. And I thank you again for your very interesting presentation. And you can provide more answers if you want to in the Q&A area.
Will do,
So,
thank you.
Thank you for your talk. So I'd like to move on now to our fourth speaker, Dr. Cecilia Arregui. She is a research associate professor at the University of Delaware. She joined the Protein Information Resource in 2005, and since then has been contributing in the areas of database curation, community annotation, text mining for biocuration and ontologies. Cecilia is here today to talk to us about scalable literature integration and biocuration at the Protein Information Resource. And with that, Cecilia, take it away.
Okay, thank you. Thank you for the invitation, and it is a pleasure to be here. Today, I will walk you through some of the efforts in integrating the literature with structured annotations in our resources. So let me move that. So I work at the Protein Information Resource. As you already know, we are part of the UniProt consortium. So you heard from Alan about UniProt. I also work in BioCreative. You heard from Lynette about that too. But I'm going to focus, Alan explained a little bit how literature mining is being integrated into the curation workflow in UniProt for the enzyme functions. I'm going to talk a little bit about adding additional information that comes from the literature to complement the information in the curated entries. And also, I'm going to highlight other projects that we have at PIR. So just very briefly, I'm going to walk you, as you know, UniProt is a resource for protein sequence and functional information. We also host the protein ontology, which is an Open Foundry ontology, which contains terms for proteoforms with the hierarchy and the accompanying annotations. And proteoforms, I mean all the products, protein products of a gene, like variants and modified forms, and so on. And then we also host, PIR hosts the IPTM-Net, which is a database for post-translational modifications. So in terms of text mining, text mining can assist in a variety of tasks, like finding the relevant publications, triage, the extraction of relevant facts. It can help in organize the publication. And the great thing is that it can run at large scale. So we can run on Medline or open access PMC, preprints, and so on. And then we can you can see the results in an integrated fashion across all Biblia. At PIR, the underlying text mining framework is called iTextMine. And iTextMine basically integrates a set of diverse text mining tools. I should say that these tools have been developed mostly by our group in collaboration with Dr. Shanker's group at University of Delaware, but also includes Pat Taylor from NCBI, you'll see later. It contains a number of interesting tools for relation extraction and of course needs entity recognition to identify entities of interest. And we have links to vocabularies, but specifically we link to UniProt for the proteins. The pipeline includes, you have an input text, which is of the PubMed and open access PMC. And it goes through a number of tools that are Dockerized and has later some post-processing to do mappings to the ontologies or vocabularies we are interested in and databases like UniProt. And then data is stored in MongoDB. We use API to then add the information, create the front end. Here is in the homepage where you can search by the specific tools, or you can search by a keyword or PMID. And the output is in a table format, and you can see also in a graph format in a network where if you click on any of these edges, it will give you the evidence source, like the sentences and the papers that where the information is coming from. iTextMine has been recently used in the context of COVID knowledge graph. It's a bioinformatics publication, and it is specifically for drug repurposing. So that's to give you a context. So the tools in iTextMine are PubTailor, like the ones for tagging, basically for name entity recognition, for tagging genes, proteins, disease, variants, and organisms. And then we have tools like RLINSP to capture information about protein phosphorylation, where you have the protein, which is for the site of phosphorylation, and the kinase when the information is available, the phosphorylation dependent protein-protein interactions. So given that the protein is phosphorylation, does it affect any protein interaction by maybe enhancing or blocking the interaction? We also capture information at the regulatory level in terms of microRNA target relations, and also what we call genomic anomalies in terms of genes that have altered expression or some mutation, how that affects drug response in cancer. So that these are the tools that currently are integrating. And here we have the numbers of publications that are touched by each of these tools. And the phosphorylation tool is the one that captures most of the data. In iTextMine, you can see the combination of results. So if you search, for example, for Veclin-1, which is a protein that is involved in autophagy, you will see a number of publications, and you can see the results here. Remember, is the only for the things that are involved in a relation. This is relation-based information. So if we search for backlink, we filter for backlink 1 here, we can see all the different other things that come out, and you can select the different publications and look at in a graphical view by visualizing the selected documents. Everything is color coded, and you can download the information. IPTM-Net is, as I mentioned, is a database for post-translation and modification, and consumes the output of iTextMine for the tools that have to do with phosphorylation. In addition, IPTM-Net, so this is the text mining part, IPTM contains information from knowledge bases like UniProt, as well as specialized information for PTMs. And it also includes the ontologies for the proteoforms to describe the different combinations in which the post-translational modifications occurs in a protein. So one of the things that I wanted to point out here is we have the text mining results, and this is related to the, I guess, to the information that the question that Kim posed to Alan. Here we include automatically information from iTextMine to IPTM-Net for the phosphorylation. So one of the things we have to do is aim at high precision to be accurate in what we include. So the way that we try to do this is by including first information about the proteins that can be mapped to UniProt. Second, by checking, because we're talking about phosphorylation sites, we can go to the sequence and check that the specific site, if it's a tyrosine or a threonine like here, we can check that this threonine is conserved in the sequence in the UniProt entry that we map to. We also provide a low confidence score to the source of Arlene's. If Arlene's P is the source, we give a low confidence score because we want to tell that this is an automatic method and could have false positives. And the number 4 here is we don't automatically include the kinases that are captured by Arlene's P, because those have the most false positive. So those are curated. Those are presented to the curator, and once curated, these are added, and the tag that showed up are called Arlene's P+. The numbers of phosphorylation sites with at least one publication in IPTM-Net are 226,000 something. The number with sites for Arlene's P is 11,000. You should know that many of the sources provide information from papers that are large scale, so that while Arlene's P will provide information for small scale papers. The number of phosphorylation that Arlene's P alone provides to IPTM-Net is 2,000. And you can see an example here where for Backlink 1, the protein we just saw, there are certain sites. This is phosphorylation of different threonines. This is a very partial snapshot, but you can see that there's this phosphorylation, this site is only provided by Arlene's P, and you can link to the sentences, and you can, when you go to the website, and see that. Moreover, you can see phosphodependent protein-protein interactions in IPTM-Net from the EFIP, where for this residue, threonine 119 of Backlink, when phosphorylated, it decreased the interaction with BCL2. And again, you can see the source. So the integration of TextMine data into IPTM-Net add experimental evidence for phosphorylation sites, in many cases of new sites. It provides kinase information. I didn't show an example of that, but it provides kinase information that is usually missing or not easily accessible in the sources, in other sources. And later also includes the functional impact of the phosphorylation on the protein-protein interaction. Now I am going to move to a different tool, which is called eMine, which was developed to capture the impact of protein coding variants on some protein property. And this is in the context of Alzheimer's disease, because this was done under an NIH supplement grant to UniProt. So the idea here is UniProt currently captures variant information with the site. This is the text format, the change, amino acid change, we have a control vocabulary for the disease, but then for anything related to functional effect of that variant, it is in free text. So the idea here is to, UniProt is moving to structure these descriptions, and we are, the eMind tool will try to do the same in a way, to extract information and present it in control vocabulary. So for example, this highlighted reduced interaction with PEN2 will be expressed with the variant in AGVS vocabulary nomenclature, and with impact with various terms from the variontology effect on protein interaction. So eMind uses a BERT-based method. We under the hypothesis that the structure and statement describing impact is independent of the specific impactee and impacted entity, we train our model on impact of microRNAs based on our myriad database. Then we evaluated the model on a smaller set that was is very small, based on like less than 100 abstracts for that were curated, and these are about Alzheimer's disease and other related dementias. We created dictionary specific for these some of the proteoforms in this disease, and we capture the impact of the protein of the variant, and also impact on other proteins. And the output of this model is made JSON format for consumption, and also we need to make it compatible for in a format for additional bibliography in UniProt pipeline. So just two comments on this. UniProt additional bibliography expands the access of published knowledge about a protein entry. It integrates the data from databases and text mining, like publications from those and annotations, and are categorized based on AppClass, the tool developed with Patrick Rouge at SIP, and that helps the navigation and discovery. So in this case, the eMind papers are integrated into UniProt additional bibliography. As you can see here, two examples, two publications that have been added. It says source eMind with a link, with a category and annotation. The annotation is a structure, but also provides the evidence sentence for such annotation. Currently, there is a small set, initial set for that has been looked at, and we are going to process the full Medline and also test on all structure annotations in UniProt. Last slide. Just lessons learned from all these activities is that it's important to have a framework that ensures that all the tools run with the same pre-processed text. When you have Medline and all, you want that all the tools that you run are on the same text. We want to light sort and view the literature. You want to be able to export in the data formats needed for the downstream pipelines. Run regular update, be able to change the tools when they are their better performance. So you want modularity, and you want to think about scalability of curation. We want to support for the development of and maintenance of the tools that are used in production, and that's for the funders. I mean, we need money for the groups that help us in maintaining those. And last, acknowledgement to the UniProt team, to the University of Delaware group that we collaborate with, and NCBI, of course, John Lewis group at SIV, Patrick Rouge group, and the NIH funders for you. Thank you.
Thank you so much. That was a really interesting talk. It does look like there's a question. I'll go ahead and read that out to you.
Okay.
It's about UniProt associated text mining approaches. So have any of these resulting text mining efforts been added to the back end of UniProt for retrieval using the retrieve ID mapping tool at the database? Or have they been integrated with those resources to extend the information being curated there?
Okay, so you can access these, like the additional bibliography information that I just showed. You can access that through the ID mapping tables that we offer in UniProt. It's under the, I don't recall what is the name, but you have the publications that are in UniProt and you have the additional publications. Those show there, but it's the PubMed ID. We also have a site, a publication, where you can search for those information. But please contact me if you want to figure out what is the use case, so we can make sure that, I don't know who asked the question, but to make sure that we can follow up and address that.
Yeah, this comes from Lindsay Anderson, and you'll be able to go into the Q&A session. And,
yeah, sorry, now I have the thing all over.
Yeah, and see that. So I have a question. So
yes.
Maybe it's not a fair question, and it's something I could have asked to all of the speakers, but how much have you ever done any kind of cost savings? Like having these curation tools enables you to do more work at scale faster. Have you ever tried to do any type of modeling in terms of how much faster, you know, what is that sort of cost benefit for having this type of tool?
We actually, within the BioCreate, I cannot tell for these particular tools, and these ones are mostly to, it helps the biocuration for UniProt in terms that you can search for candidates for curation. So that make that, like all these additional bibliography that we add with the annotations, plus we have community annotations as well. You can search on them and identify targets more quickly for annotation that contains potential valuable information for annotation. In terms of doing some analysis, I don't remember, Alan, if you can reply that, if there's anything done on the with the SIP curators. I think we had something, but I can tell from BioCreative, we did experiments, right, with the interactive task. And that was part of what we're trying to figure out, whether text mining tools have helped in making the task more efficient. And the point is we try different tools and with a small set of curators. So whatever I tell you is not going to be significant. So the only thing I can tell is that with some of the tools, we did see that it helped the curation quite a bit. With others which have usability issues or which the tools was terrible at performance, no.
Okay, well, thank you again. And I'm going to go ahead and move along to our next speaker. So thank you very much, Cecilia. So next, I'd like to introduce our last speaker for this session, which is Dr. Lars Jensen. He is a professor at the Novo Nordisk Foundation Center for Protein Research at the University of Copenhagen, where he leads a group specializing in network biology. His group develops many open tools and database resources that rely on text mining, the most famous of which is the heavily used String database. And Lars will be here today to present a talk on data and text mining of protein networks. Lars, go ahead, take it away.
Now I'm unmuted. Thank you very much. Thanks for the invitation. And it's a pleasure to present to this large group of people. That's one of the advantages of an event moving online. So I'm a bit of an outlier in this session because although I have developed some text mining tools that are targeting curators, including one that did statistically insignificantly good in the BioCreative interactive task, what I'm mainly doing is actually not making tools for curators. Instead, what I'm working on is making tools to help people who are doing big omics studies. So typically, as you're probably aware, people do nowadays lots of transcriptomic studies, proteomic studies, things like that. And the end result most of the time is a long list of proteins or genes. And the big question then is, what do we do about that? And typically what people want to understand is actually the complex interplay of the many different genes or proteins on this list. And for that, of course, protein networks are a very useful abstraction to help doing that. So for this reason, I am heavily involved and have been for many years in developing the String database. So the string database is a database of protein interactions and associations, both predicted and known, which you can look up. And when you're looking at a network querying with a protein or multiple proteins, you can look, just like Cecilia described, you can click on any one of the edges and get the underlying evidence. So what's the input for making the string database? Well, firstly, we start with 14,000 genomes in the latest version, which encode a total of 67.6 million different proteins. For those, we try to link them to each other using both physical protein interactions, so proteins physically sticking to each other, and functional associations, which are more loosely defined as just two proteins working together, for example, catalyzing subsequent steps in a metabolic pathway. There are many different evidence types from which we get these. So one is genomic context, which is simply what can you do when you have simply a pile of 14,000 genomes? And you can look at things like trying to predict operands, looking at which genes sit next to each other and are transcribed in the same direction. You can then look at whether that's evolutionarily conserved across a lot of distantly related organisms, in which case it can be a powerful signal for inferring functional associations. We, of course, also integrate all kinds of experimental data. This includes physical protein interactions and co-expression networks, many things like that. And the important thing here is that both for this and for the genomic context, we have scoring schemes that allow us to rank things from what is most reliable to what's least reliable. So we're not having sort of a certain precision cutoff. That's left to the user. We rank everything, we score everything, and it's up to you to decide what's good enough for what you're doing right now. We also, of course, are happy to take the, for us, low-hanging fruit produced by hard labor of many curators. So that includes things like computer parsable pathway databases. It could be KEGG, it could be Reactome. We take a dozen of different pathway databases and integrate them all into String. And then on top of that, of course, we want to integrate all the stuff that is known from the biomedical literature. So go through this pile of more than 10 kilometers of literature if you were to actually print it all out and put it on top of each other. However, we don't do curation, and that's an active decision. We just don't have the resources to do that, and there are other people who've been presenting in this session who are doing an excellent job at it, and we're not trying to compete with that. So what we're doing instead with this huge pile of text is, of course, automatic text mining. So the first step in automatic text mining is named entity recognition. And that's, of course, the task of recognizing stuff with names mentioned in the text. So for that, unsurprisingly, you need to use a good dictionary so that you know that you have a protein called cyclin-dependent kinase 1, you have a gene called CDC2, and in fact, CDC2 encodes cyclin-dependent kinase 1. So you need to know that these are synonyms for this purpose. You need to deal with orthographic variation, which is the fancy way of saying that people might write the same thing in slightly different ways. So you'd have, for example, from UniProt that this protein is called cyclin-dependent kinase 1, and you want to still recognize it when you go through a document and it's written with a hyphen. And lastly, you need to block bad names, because there are some names that are in your dictionary for all the right reasons. They might even be the recommended Hugo gene symbol for a certain gene. But if you go through the literature and you think that SDS is a gene, you are wrong more than 99% of the time, because most of the time people are talking about the detergent, the compound that you use for doing all kinds of different experiments in the wet lab. Once we've done the named entity recognition, and of course also identified not just that these are genes and proteins, but which genes and proteins, we do automatic relation extraction. And the simple approach to that is co-mentioning, simply saying if A is mentioned together with B, well, let's guess that A and B are doing something together. And you might argue that, well, people could easily mention two things together without them being related, but that's where we do counting. So again, we have scoring schemes, so we can calculate a sort of consensus score of, do people mention A and B together more than you would expect by random chance? The problem with this approach is, of course, it totally ignores the context. We're just looking at, here's a gene name, they're mentioned together a lot. Hey, they're probably related. Let's ignore the rest of the text. And that's why we, like everybody else in the field of text mining, have moved on to deep learning. And for that, we use pre-trained language models, and nobody is going to be surprised that we too are using BERT. More specifically, we're using BioBERT Large at the moment. And what we do is then we take this and we basically fine-tune this model for specific tasks of our interest using annotated corpora. And this can be firstly to identify bad names. So to find more names that should be blocked and not misidentified in the named entity recognition. It's also to extract interactions of specific types. So for example, we've trained models to extract physical protein interactions, but we can also have specified directionality, for example, for regulatory interactions where A regulating B is clearly not the same as B regulating A. We're doing a quite good job, I would say, nowadays with these methods of extracting what is stated in the text. And we're also able to then again, like in the co-mentioning, combine the results from multiple statements and put it together so that we can say, is there a general consensus of this being linked to that in a certain way, possibly with a direction. So that way we're able to really summarize the literature. So where do I see the big limits in this approach? Well, we're doing great. So models will keep improving, of course. We keep getting better and better models. They're bigger pre-trained models. We can download them. We can train them on bigger corpora, and we can keep getting better. And error rates will drop. And what we're seeing is that I'm pretty confident that in the next decade, we will definitely reach human level. You don't have perfect inter-annotator agreement if you have two humans go through the same text, even if they're experts. And you can't expect perfect agreement between a human and a computer model either. But we're reaching the point where it is quite remarkable how well it can be done. So that way we can do an excellent job of extracting what is being stated in the literature. But therein lies the problem, because the literature is imperfect. The literature contains false statements. That's one of the assumptions that nobody that I'm aware of really in the text mining field thinks about. When we're doing text mining, we're thinking about, can we correctly extract what's stated in the text? If we do so perfectly, they make no false positives, we have a precision of 100%. We extract correctly what's said in the text. But what if what's said in the text is not true? Computers are completely naive, so they just take everything at face value. And since we're taking everything at face value, we are then going to take every claim ever made by anyone in the literature and dump it into a database. That's clearly not what we want to do if we want to truly automate the task of doing curation. So our goal is, of course, that we want to build a knowledge graph that can help people interpret their experiments. And to do that, we need a knowledge graph that doesn't contain everything ever claimed, but rather captures what's true, what is known, what actually holds up. And for that, we're going to have to deal with the issues of not all papers are equal. So even when we're doing statistics and doing consensus, we're just counting papers or counting sentences, scoring how consistent is the literature in terms of saying the same thing. But we're still considering every single article to be equal, and everyone who's read the literature or anybody who's a curator knows that that is certainly not the case. So we need to get a computer to not just be able to read the literature, but actually be able to read the literature critically. And that's why I'm not seeing much headway being made at the moment. It's very unclear which direction to take here in terms of getting a computer to not just uncritically extract everything stated. So I was rather fast, and I just want to acknowledge a lot of people who've done the work. So the string database is a huge collaboration. It's run over many years. It all started in the group of Per Borg at EMBL, where Christian von Mehring was staff scientist when I joined the group. Christian now has his own group, also with funding from the Swiss Institute of Bioinformatics, running a big team there working on string. I have my own group in Copenhagen, funded by the Novo Nordisk Foundation, working on string, especially the text mining aspects of it. So I want to especially highlight, in addition to Per and Christian, Damian, who's a former PhD student of mine, who's now working in Christian's group, and Nadja, who's doing a lot of the work these days in my group, and also working on integrating this with tools like Cytoscape so that people can really work on big omics data sets, not just via web interface. And all the deep mining-based text mining is work done in my group by an incredibly talented postdoc, Katerina. She's funded by the EU on a Marie Curie Fellowship, and it's a collaboration with Sampo Pysalo's group from the NLP Center at the University of Turku. So with that, thanks for your attention, and I'm happy to take questions.
Thank you so much. That was a very interesting talk. I'm always impressed at the approach that you took to your slides. It's not something I can pull off. So there is a question for you. Actually, there's a couple I see coming up. One is in the Q&A area. This is from Valerie de Crecy-Lagarde. I use string a lot for teaching and research. In my experience, I have to eliminate text mining as otherwise it is noisy. So I am curious to try again with your new method. But the main issue in general is that a string does not separate paralogs, and groups often associations that are for paralogs nor orthologs together. I'm sorry, I'm having a hard time with how this is reading. Is there any idea to address this issue? So basically it's a question about orthologs versus paralogs.
Right. So I think the question, I'm not quite sure what exactly. So first of all, thanks for using string. And I think the problem of orthologs and paralogs, I don't really see that one specifically related to text mining. So one of the things that I didn't talk about is that in string, we do also transfer between organisms. So for example, if you're looking for a network in human, we take into account the evidence from model organisms and do transfer. And of course, if you're doing long distance transfers, it's not one-to-one orthology and things can get mixed up that way.
Right. So there you might be confusing paralogs with orthologs.
Well, it's rather that you don't have one ortholog, right?
Yeah.
If you've had gene duplications in one lineage, you have multiple paralogous genes that are all equal orthologs of the other gene. And then when you're transferring, it's that there's not a right answer on where to transfer it. However, we do penalize such transfers quite a lot, so that really shouldn't turn out among the high scoring interactions. When it comes to noisy with text mining, I don't know which organism you're working on, that might be helpful to know, but generally I would say they're rather unspecific because they're linking things that are mentioned a lot together in the literature, but they typically, I would say, I mean, we benchmark them the same way as everything else. So the quality, if you put a confidence cutoff, the quality should be comparable. One of the things that might interest you, and that's where the new method comes in, is that in the latest version of string, and that will also be in the future versions, instead of having these fuzzy functional associations, you can choose to have a pure physical interaction network. And in the future, we plan to expand that with other interaction types like the regulatory networks coming out of text mining and other data sources. So that definitely helps clean up things a lot there.
Okay. So there's a few more questions coming in. Do you have a knowledge graph that is shareable that you're building from this approach? This is from Amanda Hicks.
So we're talking about the text mining, I assume. So since we're currently developing the methods, we don't have a knowledge graph that is available yet, but absolutely, I mean, all the resources I'm making, not just the string database, but we have gene disease associations. We have lots of things that are based in part on text mining, and all of them are available under open licenses. And we will be doing the same with the knowledge graph when we're pulling out regulatory networks and so on. As soon as we have something that is ready for prime time, we're going to give it out under open licenses.
Okay. Another question from Dorothy Tren. Could crowd validation be employed?
We've thought about it and sort of looked at some of the statistics. One of the problems you have with crowd validation is that really most users are not going to do anything. That's the same thing you see with Wikipedia, right? The majority of people using Wikipedia never edits anything. So we've looked into it, and honestly, if we look at the time it would take to develop the whole web interface and backend infrastructure for maintaining the user fixes and carrying them over to new versions when we're updating, if we invest the same time ourselves in just trying to make the text mining better, I think we'll get more improvements out of the same time invested than if we try to make a curation scheme that allow other people to fix the errors.
Okay.
So it's not something we've prioritized so far. It's purely a matter of resources, right? You can do one or you can do the other. You only have so many people.
So
it's definitely something that could be done, but it's not something we're planning on doing.
Thank you. There's more questions. So this one comes from Lindy Anderson. How would the named entity recognition results be validated sparingly where ambiguous synonyms are cross-checked to consistent resource identifiers that are being actively curated, so that updated annotations and deprecated synonyms are being critically ingested, not to be confused with false statements when the literature evolved over time.
Right.
Deprecate, so from
Yeah,
deprecated.
so of course, names that have multiple meanings and have changed meaning over time are always a challenge. And there are many separate issues here. So there's the species disambiguation problem, where of course you might have a CDC2 in budding yeast and CDC2 in human, and it's completely unrelated genes because it's a DNA polymerase in budding yeast and it's a cyclin-dependent kinase in human. And so you need to know which organism the paper is about. And we're doing organism name recognition first to basically find out which organisms we should look for gene names from. So that helps on that side. Then what we're doing on top of it is that when we're building the dictionaries to help sort of resolve some of the worst clashes, we have prioritization of the sources of the names. So we know, for example, what is the recommended HTNC symbol versus what is some old deprecated name. And if we have something where it's clearly the recommended name for one gene and some old name for another gene, we'll assume that it's the one where it's the recommended name, rather than just being like, we don't know. So we're trying to be smart about it, but it is a very hard problem. It absolutely is.
Yeah, and sometimes an old name gets reused. Yeah, and sometimes an old name gets reused for a different gene, which really makes things complex. So
this thing, I still don't understand fully the paralog question. I'm sorry if I'm not getting it. So we do distinguish between orthologs and paralogs. Absolutely.
Okay. So we are just about at time. There were several comments and questions in the chat that you can look at and reply to during the break or during the next session, but we are out of time, and I do want to give our attendees enough time to have a little bit of a break before we begin the next session. So I want to just give a virtual round of applause again to all of the session 4 speakers. I think it was a very interesting set of presentations and topics. And yay, we had no technical glitches. And so, as the chair, I'm happy about that. So thank you again to all of the speakers, and we will be going into about a 10-minute break with the next session 5, beginning at 1:25. So thank you, everybody.